Hey everybody, today we are going to look at cell membranes. Okay, so here is a picture of a cell membrane, and I want you to pause and think about where you think the following types of membranes for transport occur. So active transport, diffusion, and facilitated diffusion. So if you pick this one as active transport, you are correct, and we'll talk about that later. And then this one is facilitated diffusion, and then this one actually is also active transport. If it just goes through the cell membrane, that would be regular diffusion. So there's lots of proteins that are embedded in the membrane, two types. There's peripheral proteins, which would periphery, like your peripheral nervous system is in your arms and legs. And so that would be like these blue circled things. And so they're bound to the surface of the membrane. Their function is either recognition or cell-to-cell communication. And we want to think about hydrophilic. So they're hydrophilic because remember the heads of the cell membrane are hydrophilic. And so the hydrophilic proteins are going to interact with the hydrophilic heads. And remember, outside of the cell is lots of water. So that's why they have to be hydrophilic. The second type of protein is integral proteins. And so those span the distance of the whole membrane. So they have to have a hydrophilic on the outside and a hydrophobic region on the inside. And an example would be these transmembrane proteins. The other thing that is in here are cholesterol. And so cholesterol is the red circled thing right here. And those help with fluidity of the membrane. Remember the fluid mosaic in different types of environments. And then the blue circled things are carbohydrates. So these right here are carbohydrates like glycoproteins and glycolipids. And those are, their function is cell markers. So glyco, remember, is sugar. So this is sugar attached to a protein, or you have a sugar attached to a lipid. So a big thing with cell membranes is the net movement of a substance from a region where it's more concentrated to one where it's less concentrated. And so things always move down their gradient. Net movement is important because substances are always going back and forth if they can go through the cell membrane, but the net or the majority of the movement is going from a high concentration to a low concentration. If it's doing this, it's passive transport, which means they don't require any energy, and examples are simple and facilitated diffusion. Okay, so a couple types, remove that. A couple types of endocytosis. So endo means inner, cyto means cell, is phagocytosis or cellular eating. And that's where a cell, down here you can see the example, a cell engulfs a solid particle. It could be, it could be a bacteria, it could just be a particle. And so it engulfs it, and a lot of times, or a virus, and a lot of times it'll put it into like a lysosome, and so that it can digest the particle. Penocytosis, which is right here, is cellular drinking. So molecules are taken up, large amounts of water, and molecules are taken up into these tiny vesicles. So like if you have solutes or stuff on the outside. Receptor-mediated endocytosis is where a specific molecule, like these little stars, bind to these receptors, and these little stars, if they bind to receptors, are called ligands, and that triggers a vesicle to form. Okay, so think about how things can go across the membrane. There's 5 of them. Okay, so the first one is simple diffusion, and so that's from high to low concentration, and the molecules that can do that are small and uncharged, like oxygen and carbon dioxide, and that does not require any energy. So those go straight through the membrane. The next one is facilitated diffusion. Again, high to low concentration. The molecules are bigger and they may be charged. Still no energy is required, but they do have a protein that they go through, and glucose is an example of that. Active transport, so here active means energy required, so ATP. And what's happening is it's more like a pump. And so the transport protein is going against the concentration gradient from low to high. Endocytosis is where you're bringing in large molecules, like we just talked about, proteins and larger. And of course, if you're bringing in large molecules, that requires energy. And the last one is exocytosis, which is secreting proteins outside of the cell, proteins and larger, and that also requires energy. So for example, hormones that are made in the pancreas, like insulin, are secreted outside of the cell with exocytosis to trigger a response in other cells. Here's an example of endocytosis. Here's your plasma membrane, and it's taking up cholesterol, so you can see the little vesicles starting to form, and then the cholesterol's inside the cell. Here is another example of endocytosis. Pretty cool. This is a macrophage, so part of your white blood cell, and it is engulfing a parasite. So exocytosis is where the molecules inside of the cell are going to be secreted. So they're larger, they're proteins and bigger, like hormones and waste. And so here you have a vesicle, and it's now fusing with the membrane because look, it's made of the same exact molecules, phospholipid bilayer, so it can just fuse with the membrane. So what factors affect diffusion rate? How fast things diffuse, and how do they do it? Maybe. There we go. So steepness of concentration gradient. So the steeper the gradient, so if you have a ton of stuff on one side and very little on the other side, it's going to diffuse faster. Molecular size, so small molecules can diffuse faster than large molecules. So small molecules take less energy to move, and so they are going to diffuse quicker, and you'll play around with a simulation for that. And temperature, so the higher the temperature, right? So if the molecules are moving faster, then you're going to get faster diffusion. So osmosis is the diffusion of water molecules across a selectively permeable membrane. So the direction of net flow is determined by the water concentration gradient. So as you can see in this picture, your water molecules are little and white, and your protein molecules are too big to pass through the membrane. And so the net flow is this big arrow, if you see it in the background, and it's going to go from where you have the most water to where you have the least water. However, if you notice, there's still molecules that are going to go, water molecules that are going to go this way, but the net movement is to the right. So the side with the most solute, things that are dissolved in water and have the lowest water concentration, and then water is going to move from its highest concentration to its lower. So it's opposite of, if you have lots of solute, then you're going to have very little water, and so it's opposite compared to the solute concentration. So here is a YouTube like that diffusion and osmosis challenge we did. So the sugar molecules can't move across the selectively permeable membrane, but the water can. So if you notice, the water is going to move from left to right, as shown here, because you have more solute and less water over here. So the water is going to balance out so that the concentration of the solute is the same. And here is a picture that shows the waters going back and forth. The solutes cannot, and so the water is going to net movement, because there's three arrows, is going to be to the right. Now water is a polar molecule, so lots of it cannot get through the membrane by itself, so there are things called aquaporins. or aquapores. And so you can see here that they are just, it's a form of facilitated diffusion that allows, it's a pore, basically a hole that allows water to go in into or out of the cell. So since it's polar, it can't go through this hydrophobic area in the middle. So how do cells balance their water? So tonicity refers to the relative solute concentration of two fluids separated by a membrane. So cells have a tonicity compared to the outside environment. So if a cell is hypotonic, what that means is there are fewer solutes. So here's an example. There are fewer solutes outside of the cell, which means there's more water outside of the cell. And that means if there's more water outside the cell than inside, then the water net flow is going to go into the cell. So I always remember hypotonic as hippo. So cells go from get bigger as a hypotonic. Hypertonic has more solutes outside of the cell. And so there's more water inside of the cell, and the cells are going to go from inside of the cell to outside of the cell. And I always think of hyper people always moving around, and so they're going to be skinny. And then isotonic is the same amount, and so the net movement there won't be any net movement, and the water will flow in and out of the cell equally. So the solute concentration and the water concentration are inversely related. So if you see here, this is some red blood cells. And so if you put red blood cells in distilled water, then that means that they are going to have a net flow of water into the cell. And so they actually would explode. If you put red blood cells into a solution of sucrose, 10%, you can see that they lose a lot of water. And then if it's 2%, then they're going to stay the same. So this one would be hypotonic because water moves into the cell and there's less solutes outside, so more water outside. This one is hypertonic, which means that there's more solutes outside, and so water is going to go from its high concentration inside the cell to outside the cell. And then this one is isotonic. So what do little organisms do when they live in a hypotonic environment, like in a pond or a stream or a lake or something like that, where there's lots of water and less solutes outside of the cell? And so this is a little paramecium, and what is going to happen is there's going to be a constant flow of water into the cell. And so in order for the cell to survive, it has to somehow get rid of it. So they have what's called a contractile vacuole. And it fills up with water, and then the cell pumps it out. So here's just a picture. Hopefully this will work. A little video on how a contractile vacuole actually deals with a hypotonic environment.
The contractile vacuoles of ciliates are permanent structures that periodically squeeze excess water out through a tiny pore. In some paramecium species, the contractile vacuoles have conspicuous feeder arms, but in most ciliates, the canal system is less apparent, and the contractile vacuole appears as a simple sphere.
Okay, and that's it for cell membranes. Have a great day.